First of all, I'd like to thank Little Things for hosting us as always. They do an excellent job. Please buy lots of alcohol and tip your bartenders. So remind everybody the format of the talk, if you haven't been here before, we're going to have a 30-minute talk and a 10-minute break to give refreshments. Please hold your questions after the 10-minute break. We'll come back and Dr. Mills will answer everybody's questions. Yeah, everybody's. So we have a few plugs to give. A few plugs first. Liam, you want to come up here? So Liam is going to tell us about the new organization in Athens, Science, Tales, and Trails and Tales.
How's it going, guys? So my name is Liam. I'm with Science for Georgia, and we're an Atlanta-based nonprofit that has a very similar mission to the Alliance here, the Science Alliance in Athens, in terms of bridging the gap between the scientific community and the public and increasing sort of public engagement. And one of our events that we run in Atlanta, we're sort of installing, or trying to do an installment here. It's called Tales and Trails, and we just gather together. It's very casual, walk amongst one of the green spaces locally, and talk about all things like science and STEM. This month, or this next coming one, is going to be December 7th at Sandy Creek from 12 to 2. We're going to walk along the Greenway Trail for a bit, and we'll be talking with Dr. Michelle Momini about fungi and research in disease. So if you have any questions about the event or the Science of Georgia in general, you can find me after. There's little quarter flyers I can intersperse about. Feel free to take them. If you want one, I have some too. So thanks, y'all. Thank you, guys.
Just a couple more announcements. So if you check your seat, we have a flyer for the UGA Library's Capturing Science Contest. Explain the STEM concept and win $3,000 in prizes. That is still going on until December 2nd. The UGA Libraries help us out a lot. Also on your seat, there are little cards, surveys for Athens Science Cafe. It helps us improve the cafe. It gives us feedback for our funding agencies. Please fill them out. There is a box in the back that is nicely decorated, or leave them on your seat and I'll get them after. Other than that, our next cafe is right here, December 12th. Michelle, the graduate student in genetics, is going to be giving a talk on mental health in graduate school. So come on out, support her, and once again, tip your bartenders. So I'm happy to introduce Dr. David Mills from UC Davis. He is the Shields Endowed Chair, and he is a full professor in food science and technology and viticulture and enology, and he's going to give us a great talk on breast milk.
Thank you. It's an honor to be invited, and this is the first time I've given my talk about breast milk while holding a beer. I'm actually in two departments at UC Davis. I'm in the wine department, the viticulture and enology department, and I'm in the food science department. I used to work a lot on wine. I could give a wine talk if you guys would be interested in that too. But mostly what I do is work on human milk and milk in general. And so I like to say, as I said in the talk earlier today, that I work on the two greatest beverages that humans can drink. That is, of course, breast milk and wine. So I'm going to talk really about how amazing human milk is. And it's something, it's an immensely understudied fluid. And there's some just amazing, wonderful things that we're finding out about human milk and how it guides the nutrition, the protection, the microbiome development that I'll talk about today of an infant. And as we start to learn more and more, it's as if we're just scratching the surface on what is actually in milk, what evolved to be in milk. And I hope by the end of the evening you'll be convinced that we all should convince NIH to fund a lot more in human milk research. So one of the things to think about with human milk is it as a food, as a nutrition source, right? And if you think about what foods evolved to make humans healthy. Now, growing up, I looked at those three foods that are up there now, and I know that they make me healthy. I don't necessarily like to eat them. I actually like it more now than I did when I was a child. But as much as those foods are good for you, they're good to eat, they're healthy for humans to eat, they themselves didn't co-evolve with us to make us healthy. There really is only one food that did that, and that, of course, is breast milk. And breast milk, if you think about it, co-evolved with mammals, with humans, but mammals before that. And it's part of just an astounding process that is lactation. And lactation is an evolutionary experiment that resulted in the human milk we have today, or the bovine milk we have that obviously is meant for bovines, or you name the mammal. It's an amazing process that went through this incredible filter. It's an evolutionary filter that's kind of fun to think about. So there are opposing forces on the evolution of milk. And this is called parent-offspring conflict that's indicated in the slide. If you think about it, every bit of energy the mother gives away is at risk to the mother. So evolutionarily, if she gives away too much energy, she dies. Every bit of protection and or nutrition that the child does not get is at risk to the child. And the child dies, and then the mother does not pass on their genes, so that line stops. So we have opposing evolutionary forces on this fluid. You can imagine, therefore, anything that was superfluous, that evolved into this fluid but really wasn't used by the infant, would be called out immediately through evolutionary process, through selection. And you can imagine the flip side of that. It would be great to make each molecule do 15 things. Because if you make each molecule do 15 things, you make it once, you got 15 functions. And it turns out that latter story is what we're finding over and over and over again with the molecules in milk. What human milk is this amazing constellation of different biofunctional molecules that are both nutritional, bioactive, aimed at microbes in some cases. It's an amazing fluid, and it's one that I think can teach us lessons about what is a functional food. Functional foods is a catchword used by the food industry now to try to make us all healthier by what we eat. And it's a good goal. We should eat better foods. We should eat more kale. But we want to understand those foods better and try to understand their mechanism of action. I think we can learn that from studying human milk. So the origins of milk, as some of you may or may not know, actually started in egg-laying animals at the time, but went through mammalian evolution. And when folks have started sequencing the genomes of different animals that are part of the mammalian evolutionary path, one of the things they noticed right away was that the genes, the proteins for milk production, are some of the most conserved among those mammalian genomes. So other stuff or diversified within those genomes, but nature kept those genes very conserved throughout this whole process. So there's something about the composition of milk that there's a strong interest to keep it the same and not have it vary as much as other animal functions might have varied. So if you think of this process, this opposing forces shaping the constituency of human milk, and you ask, well, what's in milk? And what are the bioactive molecules that are in milk? Well, there's some obvious ones that we already know. Water is the big part of any milk, of course. There's a variety of macro and micronutrients. Most of that is lactose. Of course, lactose is used to feed the neonate. Lactose is a disaccharide, and it's the sugar that feeds the neonate. It's in a sense a slow-release sugar that feeds the neonate. Fat, lipids, is a huge component of milk, and that also feeds the neonate as well. There's a variety of proteins in milk. Those proteins are both nutritional and bioactive. So some of them might have functions, some of them might be nutritional, and as I'm going to tell you, they're almost all both. And then there's this fiber in human milk, and this is what that word HMO stands for there. It's human milk oligosaccharides. This is something that people didn't really realize. Human milk, the third largest constituent in human milk is actually fiber. And that fiber is non-digestible by the infant. So let's think through that evolutionary process again. 120 million, depending on who you talk to, 300 million years of mammalian evolution, resulting in a food, the third largest constituent of which cannot be used as an infant. That's a lot of evolutionary energy to invest in a constituent that the infant's actually not consuming. So why would it be there? And that was the puzzle that we and others have been trying to examine for about the last 20 years. I'm part of a large group, and thankfully part of a large group at UC Davis, most notably with Bruce Sherman, who's a very famous milk scientist, and Carlito Labria, who's a distinguished professor of glycomics at UC Davis, and we've been working on this for about 20 years now. So most of what's delivered, of course, is used as food for the infant. That makes sense. Moms are delivering nutrients. But there's some interesting stories to tell about some of those molecules. And these stories help me amplify this concept that you might be able to make something once but have multiple activities for it. One of the more famous is alpha-lactalbumin. Alpha-lactalbumin is a very popular or populous protein in human milk. And it was always thought to be just broken down into amino acids and peptides, and the infant consumed those, and that was it. So it was used as food, and everybody thought that's what it was. And then a woman by the name of Katerina Svenvold, I'll show her a picture in the next slide, was doing some cancer research, and they were studying, in a sense, transformed cells, and they were using milk as a control, and the milk kept killing the cancer cells. We couldn't figure out why that was doing that. And it turns out alpha-lactalbumin, as indicated on the left, when it goes into the infant's stomach, there's acid in the infant's stomach, it opens up, it loses a calcium, it takes in a fatty acid, an oleic acid, and it refolds to a different shape. When it has that different shape, now it's a bioactive protein. It goes down the rest of the GI tract of the infant, and what it does is what she published back in 1995. It encourages apoptosis in virally infected cells. Infants are very naive. They're infected by all sorts of viruses. When viruses normally infect our cells, one of the defenses we have to viral infection is the cell goes through a process of apoptosis, and in a sense commits suicide. And the viruses are smart, and they put a block on that. This protein unblocks that. And so it basically allows those cells that should go through apoptosis to get rid of the virally infected cells in your intestine. And an infant's intestine is particularly naive. So it's particularly at risk for viral infections. So we have, in a sense, an anti-cancer protein that, when you think about how it evolved to end up in milk, it's very strange. Its activity happens outside the host that makes it, and it has to go through a GI tract to be activated. So evolution is a very strange process that created this molecule that's only active after it goes through a baby's stomach. Not surprisingly, it turned into a cancer treatment. And so now you can isolate this molecule from bovine milk as well. And it's a cancer treatment. But it has another function. These same Hamlet molecules, they call it Hamlet because it's human alpha, let's see if I remember this right. Alpha-lactalbumin lethal against tumors. Yeah, LD for lethal. There isn't an E word. Lethal. And so this is actually they're using it to treat warts and some other virally infected cells as well. It has not just that activity, it's also antimicrobial. So now it kills virally infected cells, but it also kills bacteria. And I think they looked at it, and also kills proteins as well. Or excuse me, fungus as well. So this is an example of making a molecule once and having a huge number of activities, and that's one of the things that we keep seeing in milk. A second similar story is lactoferrin. And lactoferrin is a well-known milk protein. It has a variety of antimicrobial activities, and so that's what it's most studied for. People think it has pro-microbial activity, so they think the oligosaccharides that are stuck to it can act as a prebiotic in a sense to grow certain bacteria. But of course, it goes through the infant's stomach, it gets digested, a peptide gets released, and it makes another peptide. It's called lactoferricin. So there's another different transformation of the protein that happens inside the child. So evolution created something that had to be activated outside the host again. For its activity. Now this one modifies the gut microbiome because it kills bacteria. So why would nature, in the form of milk, want to control the gut microbiome? Well, you guys have probably heard about the gut microbiome already. There's certainly microbiome science. We are covered in bacteria. They're everywhere. They're on us and inside of us, and they're important for our development as animals. They're extremely high concentration in our GI tract. They perform a lot of the digestive functions that we can't, particularly in our colon. And setting up that organ is a complex process, and one that's not well understood. And as more and more people study the development of the microbiome in any animal, but particularly humans, they're starting to recognize that the early development of that microbiome and perhaps dysbiosis or the wrong trajectory in that microbiome can lead to all sorts of be associated with all sorts of downstream problems, malnutrition, stunting. Some people are talking about allergies and atopy, eczema, even autism is people are hand-waving that towards the first year of life for the development of the microbiome. So we're very interested in that aspect. So you have to remember, as an infant's born, you're, for the most part, sterile. And so the infant is, in a sense, this big magnet for bacteria. And as soon as you're born, you become colonized by bacteria immediately. Even if you're in a hospital and they're stripping you right away, you're being colonized dramatically and quickly. A guy by the name of Jeff Gordon did a study of mice that you could make germ-free. And he tested out, if I take microbes from the soil and I put them into a germ-free mouse, will they live in the gut of a germ-free mouse? Sure enough, they did. And he took microbiota from a fish and put it in a germ-free mouse. And sure enough, that population rearranged, but it grew. So a gut of an animal can grow lots of things. That doesn't mean it's the healthiest in that animal, but it's a rich environment to grow stuff. And so milk is there to shape that, in a sense, shape that gut microbiome development that is now believed to be so important for later life. So how might it do that? Well, I've already talked about two proteins that are antimicrobial. So there's all sorts of suppressive ways that you might knock down populations of bacteria. One of the things that's delivered in milk is immunoglobulin. So that's antibodies. And of course they bind to bad bacteria and flush them out. So that's another way. Now, do we have a way of enriching good bacteria? How might that happen? Well, let's go back to that fiber that's in them. This fiber is composed of sugar linkages that are not digested by the infant. So the infant doesn't produce enzymes to break them apart. And if you look at those structures, those are just different cartoon structures of the kinds of glycans that are actually in human milk. You need an enzyme to break down every one of those linkages. But humans are not producing it. So the infant can't break it down, and it will flow right through the infant. And babies that don't have the right bugs in them have these things flow right through them. So the question is, does it really enrich a specific microbiome? There's a lot of different degrees of polymerization that are made of human milk oligosaccharides, and there are lots of different structures. And it's very complex, and it's very hard to study. You use very high-end mass spec to be able to differentiate this stuff. And the human milk oligosaccharides have been studied for their prebiotic behavior that I'm talking about, this kind of behavior. But of course they have, so far, 7 different functions. I think. So one function is enriching the good bugs. Another function is deflecting the bad bugs because they look like the cell surface of the intestine. And so a bad bug thinking it's binding the intestine is actually binding milk saccharide that gets flushed out. And then there's all sorts of direct effects on the host, and there's more coming. And there's a huge industry now to make these oligosaccharides synthetically. First and foremost, people want to add them, of course, to formula. But all the people who are making them are recognizing that there's big effects on adults, GI tracts. And part of the reason is this multiplicity of effects. And this is just really the wonder of milk being something like this. And in infants, in human infants, there's always a connection of breast milk with brain development. And people have been trying to find what is the component of human milk that's linked to brain development. There's a lot of thought about silated oligosaccharides. And the other debate people have is that human milk oligosaccharides are dominated by what we call fucosylated oligosaccharides. So they have a fucose molecule attached to them. It's one of the sugars. And humans are unusually highly represented in fucosylated oligosaccharides by comparison to any other animal, far more than any other animal. And the thought is that maybe that's related to brain development in infants. We have the largest brains, and so maybe that was an adaptation and change in human milk that allowed, in a sense, the growth of a very rapid growth of an infant brain. And that's still being debated hotly. Okay, I'm going to talk about the prebiotic behavior. And so I'm going to define some terms that you might have seen before. You guys have heard, I'm sure, of probiotics. Probiotics you can buy at the grocery store. They're in your average yogurt. Those are live organisms. There's a definition to this. There's live organisms that, when given in adequate amounts, provide some sort of health benefits. So usually these are the bacillus or bifidobacteria that you can find in just about any yogurt today. Of course, there's a big probiotic market just to sell you probiotics in a pill as well. Prebiotics are undigestible or non-digestible fiber that makes it down to your colon and enriches a beneficial consortium of microbes in your colon. And it's sort of a saccharolytic colon. And that produces short-chain fatty acids in your colon, and that's beneficial to health in a whole bunch of different ways, as well as for peristalsis and other aspects. And then when you combine them, probiotics and prebiotics, they call it symbiotics. These are some of the terms you might see in some of the products today. Well, one of the things you can imagine is that these human milk oligosaccharides are these really complex structures. For a bacterium to eat this, you have to have all the enzymes necessary to break all these structures down, which means you need a repertoire of 15 different enzymes to sort of go at it. And not every bacterium will have that. So it is a good likelihood that this is going to enrich a certain type of bacteria that could actually grow on it. Now, is there evidence that the microbiome of kids change, or there is a specific milk-associated microbiome? And if you've had children, and I haven't personally had children, but I've been associated with the process, and one of the things you know is that whenever your child squeals,